disorders. And because it's a 30-minute talk, I was just going to pick on one or two. And start with this vast calcium signaling toolkit that Michael Berridge has put together. Obviously, he's touched all of our lives in many ways, and he's dearly missed, so I thought this would be an appropriate way to start. And this toolkit is by no means complete, but I think it makes the point that cellular calcium signaling is intimately tied to so many essential cascades and functions that if it's dysregulated, it can invariably lead to various forms of pathology and cell death. And this is becoming increasingly apparent in neurological disorders, certainly in Alzheimer's disease, but also in Huntington's disease, incorporating work by Ilya Brezprezvani and others, frontotemporal dementia, Parkinson's disease, work by Vicky Bolotini and others, and then traumatic brain injury, which is an area that we more recently started moving into and looking at cellular signaling abnormalities occurring from traumatic brain injury. So that's what I'll be incorporating a bit, talking about later on. But just to sort of refresh and go over some of the key calcium channels that we've been focusing on that seem to be primary in driving some of the early stages of Alzheimer's pathophysiology, seems to largely come from the ER stores. Both IP3 receptors and ryanodine receptor dysfunction have been implicated. We focused primarily on ryanodine receptors in large part because of its intimate role in synaptic signaling and its disruption out in synaptic compartments, which, because of this learning and memory implications, has, I think, a strategic role as a therapeutic target. This excess calcium from the ER is also driving mitochondrial reactive oxidase species generation, as well as defects in lysosome and autophagosome handling. So this all ties into the metabolic stress and protein mishandling that we see common in many neurodegenerative disorders. And much of this can be tied back to defects in calcium handling further upstream. And to summarize, years of work, not just by myself, but by many others, is starting with this slide, the early manifestation of ER calcium dysregulation in Alzheimer's mouse models. And what we've identified is that, in the soma, we're seeing, say, a 2 to 3-fold increase in ryanodine receptor evoked calcium responses. And this is tied to a variety of different cell signaling cascades. As it pertains to neuropathology, it can upregulate kinases that are driving the hyperphosphorylation of tau that leads to those neurofibrillary tangles. It's upregulating enzymes that are cleaving amyloid precursor protein, or APP, such as BACE, and this leads to increased pathogenic species of A beta 42. So this is what's forming those sticky plaques in the brain. It's also increasing and altering gene transcription pathways, as well as post-translational protein modification. So these are all kind of, a very brief summary of some of the calcium-regulated signaling cascades that go awry in the soma. But if we move out to the spines, where the memory encoding is first occurring, we're seeing this massive, massive increase in ryanodine receptor evoked calcium release. Normally, it's really playing a minor role, only under very strong stimulation paradigms. But in these AD mouse models, it's not only altering like basic neurotransmission properties, which we know is, calcium signaling is fundamental for basic neurotransmission, basic synaptic plasticity. It's this plasticity that's driving memory encoding. And it's also altering the structure of these dendrites with the microtubules and the actin cytoskeleton is also regulated through calcium signaling. And we're seeing this loss of synaptic structure in addition to the synaptic function. And if we can normalize this calcium, all of these synaptic signaling deficits and structural deficits go away. Again, so this is years of work by many, many labs, but it's, again, pointing to the central role of early upstream calcium driving a very diverse set of downstream cellular pathological phenotypes. And I highlighted earlier or mentioned that this is all done in transgenic mouse models, which for coming up on decades now has been the primary source to really get at nuts and bolts of neurodegenerative and AD pathology. It's just particularly, say, in Alzheimer's, in most cases of Parkinson's, it's sporadic Alzheimer's. You can't really generate a mouse model or an animal model of it. You need to generate these genetic formulations. And you can get these very profound phenotypes by inserting human mutations that will cause very rare forms of these diseases, not necessarily representative of the sporadic forms, when it generates this very profound phenotype. However, as we're now moving to phase 3 clinical trials targeting many of the histopathological features of the disease, such as beta amyloid and APP processing, what worked incredibly well in a mouse model, failed consistently over and over again in human trials, so suggesting there's some discrepancy or difference in the basic mechanisms between the humans and the mouse. So more recently, in collaboration with my colleague Bob Marr, also in the neuroscience department at Rosalind Franklin, working with human neurons derived from human patients. So age controls, human AD patients, sporadic and familial forms. And he can take these fibroblasts and redirect them to either an iPSC, an inducible pluripotent stem cell, and then redirect them into a neuron, or even going directly from a fibroblast directly to a neuron. And there's positives and negatives to doing it both ways. But the point is he can do this successfully. So if you give me a neuron in a dish, then I do what I do, or as my comfort zone is start poking it with electrodes and start filling it with indicators so I can now study neurophysiological properties in these neurons. And so we can just demonstrating here, we can generate mature action potentials, generate sodium and potassium currents. And importantly, this is a whole cell patch lamp recording showing spontaneous excitatory postsynaptic currents. So because they're CNS sensitive, we know that they're glutamatergic and they express AMPA receptors. And we see these both in the age patients and then the AD patients as well. So we have all the basic architecture to work with in terms of looking at neurophysiology and neurosignaling. And of course, my very first burning question was, what do these calcium signals look like, right? Because if there's no difference, then I really need to go back to a different drawing board. And across a variety of different AD lines, we consistently see this increase in ryanodine receptor evoked calcium signals compared to the age-matched, non-AD patients. And then this is sensitive to dantrolene. So we can basically normalize this excess calcium response by targeting the ryanodine receptor channels and then also other dantrolene analogs. So at least we feel we've got a basic similarity between what we've been looking at in the mouse model in extreme depth to just the first pass look at altered calcium signaling in the human neurons. And then the next step is, well, one of the main features of Alzheimer's disease is that A beta 42, that pathogenic species resulting from APP cleavage. And what we see, these are in black here is all the Alzheimer's neurons, and they're generating increased levels of A beta 42. So it's the stickiest form of that peptide. This is our age-matched control here. What I found particularly interesting is that we've demonstrated this in mouse models, but if we incubate these Alzheimer's neurons in a dantrolene derivative, a more soluble form of dantrolene, we now reduce this A beta 42 level back to a non-AD level, right? So again, pointing to the important role of altered calcium signaling driving miscleavage of APP into A beta 42 in a pathogenic form. Another major feature of Alzheimer's again is that hyperphosphorylated tau, which we're seeing in panels A, B, and C. These are the AD neurons and standing for one of the species of hyperphosphorylated tau that we're really not seeing any of that in the non-AD patients. And I think we're still ongoing studies to show that we can prevent this with that ryanodex or dantrolene treatment. We've shown it extensively in mouse models, so we're hoping to see the same thing in the human neurons, or otherwise certainly expect to. And if not, then we'll figure out why. Part of the reason we think this is occurring is I mentioned some of the lysosome autophagosome interactions with calcium signaling. And I know I've shown some of this data at one of the more recent meetings, but again, looking at the rate or amount of autophagic flux or the accumulation of these autophagosomes that normally should be digested by lysosomes, right? So these autophagosomes are clearing away undesirable proteins such as A beta FASO2, such as hyperphosphorylated tau. Then they attach to and get digested by the lysosomes to clear these unwanted protein and sort of refuse products in the cell. And what we're seeing in the AD neurons here is that these autophagosomes aren't being digested. They're accumulating. And we also know that these lysosomes, their pH is intimately affected by calcium levels being released from the ER as well. So it seems to be a sort of a multifaceted loop between excess ER calcium release, interference with lysosome function, and then the accumulation of these autophagosomes that we know are harboring increased phospho-tau. Okay. So again, I just sort of pointed to some of the major features of which upstream calcium drive Alzheimer's disease, but it forms a feed-forward cycle of pathology, and how it can sustain itself. I just showed some examples here where, yes, that increased calcium, which is facilitating A beta generation, can occur, but likewise that those increased beta amyloid peptides can facilitate calcium signaling. So whether it really is A beta that's initiating the disease or ryanodine calcium signaling or some other factor, once you invoke this cycle, it's capable of sustaining itself. So these A beta 42 peptides, one can generate these calcium permeable pores, and also induce increased intracellular calcium, which again leads to increased A beta 42, and around we go. Similarly, with hyperphosphorylated tau, we know that this increased calcium is potentiating the phosphorylation of tau. It's also upregulating the activity of several kinases, such as GSK3 beta, CDK5, MARC-PAR1, protein kinase C. All of these mediate the phosphorylation of tau. So again, this is kind of how it's moving forward. But this phospho-tau also can result in increased intracellular calcium, this cholinergic mechanism as well. So again, it becomes a feed-forward cascade, but this isn't necessarily specific to Alzheimer's per se, by any stretch. So again, I mentioned certainly in a variety of different neurodegenerative disorders, there's evidence of calcium dyshomeostasis. And what can and does this dyshomeostasis trigger or invoke these same pathways, or is there something specific to each of these conditions? And that's sort of more of an overarching question I've been thinking about more and more is, we're seeing sort of this non-specific increase in ER calcium release. Why aren't we getting Parkinson's, or why aren't we getting Huntington's? Why is it Alzheimer's? What else is, what are the specific mechanisms that are being triggered? And one of the closely related components of Alzheimer's disease is, well, age is being the #1 risk factor. And that's far and beyond for sporadic Alzheimer's disease. But the next closest, sort of environmental risk factor is previous head injury. And traumatic brain injury is a much more less specific type of neurological condition, but it shares many of the same histopathological features. So APP dysregulation, hyperphosphorylated tau, cognitive deficits, a variety of signaling alterations. And the thing is, with traumatic brain injury, it's fairly easily modeled in the lab, unlike sporadic Alzheimer's disease or sporadic Parkinson's disease. So we've started working with a group at DePaul University. This woman, Dorothy Kozlowski, has expertise in developing traumatic brain injury models. And she developed this concussive closed head impact model. And we've used this in both mice and rat models. And you basically just have a pressurized piston and a pump, and you can administer, under very controlled conditions, an isolated, concussive impact of varying intensity. So we're trying to model sort of a mild to moderate concussion, something that would be not unheard of by many of us, either in sports or even, auto accidents. Many of us, if we haven't already had some sort of mild to moderate concussion, know people that have. And something that's increasingly a topic of conversation, particularly with professional sports, is this repeat and multiple concussions. And these are particularly damaging in that, the length of time it takes to heal from a concussion still isn't entirely clear. And then if you have multiple ones after that, you really are not healing well. You have a population of already vulnerable neurons. are becoming re-injured. It's found that this leads to much longer term deficits and a higher increase in developing neurodegenerative disease, particularly Alzheimer's-like, Parkinson's-like, or something else that is I think increasingly in the news is a condition called chronic traumatic encephalopathy, or CTE. And that is largely a tauopathy. It's resulting from this accumulation of insoluble hyperphosphorylated tau. So just to show you sort of what this looks like at both gross and cellular examination. So on the left is this healthy 65-year-old brain, and the staining is fairly clear. I believe this is just a Nissl stain, compared to a football player. And I believe this is American football, but there's still certainly with European football, rugby, any condition or sport or activity where there's sort of multiple concussive or head contacts. This is a case of this, you can see this profound accumulation of hyperphosphorylated tau and protein aggregates in this brain. So, and it's not reversible. So we are interested in, what are some of the common underlying cellular mechanisms that might be shared between Alzheimer's and traumatic brain injury, and being that previous head injury is one of the leading risk factors for Alzheimer's disease. What are the sustained effects? There's a lot of research around sort of acute effects after a brain injury. We know that there's swelling. We know that there's excitotoxicity. We know that glutamatergic NMDA, calcium is increased. And, those are all points, very important cellular mechanisms to understand for, say, an acute intervention. We're interested, what are the sustained effects? How is it that these previous head injuries that months to years later can now manifest in Alzheimer's like dementia or pathology? So we were looking at a model, both single traumatic brain injury, single TBI, or repeat TBI, where we give 3 mild to moderate concussive injuries, 2 days apart. And then after the last injury, we look 30 days later, and now we're looking at a series of synaptic plasticity, synaptic transmission, and calcium signaling alterations that can occur. And this is a fairly new, at least, field in terms of looking at these sustained effects, particularly with repeat TBI. So one of the first pretty these eye-opening phenotypes was the change in basic synaptic strength. So here we're making hippocampal slice preparations. We're stimulating these CA3 afferents that terminate in the CA1 subfield and record extracellular field potentials after stimulating these afferents. And we're just looking at levels of synaptic strength. And whether it's a single or a repeat TBI, we're seeing this acute or the sustained hyperexcitability in this pathway. One might think, okay, that's, there's a damaged pathway and maybe there should be reduced or impaired or blunted synaptic signaling. But here, we've looked at this both in mouse models and in rat models rather extensively, just to make sure that this phenotype, was consistent. And it's this very profound hyperexcitability at this synapse. We're not seeing major differences in long-term potentiation. So this doesn't necessarily manifest in single cell long-term plasticity changes, but this circuit seems to be hyperexcitable. And we're still trying to investigate why, whether it's an increase in AMPA receptor expression, a reduction in GABAergic or inhibitory excitatory balance. But again, it was a very consistent outcome. And since it's the calcium signaling talk, we certainly focused heavily on what is the intracellular calcium signaling deficits that may or may not be present. And the first thing we typically look at is voltage-gated calcium entry. So this is using patch clamp recordings in a single CA1 hippocampal neuron, and we inject depolarizing current and can trigger trains of action potentials. And unlike Alzheimer's disease, in our TBI models, we're seeing a significant increase in the amount of calcium coming in through these voltage-gated calcium channels, which again, it was struck us as unusual because we never see this in the Alzheimer's models. So what we were hoping to find perhaps is these very parallel similarities. This was a clear point of divergence. But I've also been thinking this is this is a phenotype typical of aging. So in aged mouse and rat models, this is work done by Phil Landfield and Olivier Thibault and others, that with aging, you're seeing this increased conductance through individual L-type voltage-gated channels as a function of aging, irrespective of Alzheimer's or any neurodegenerative disorder. So, and it said that's happening at the single channel level. It's not that there's more L-type channels, it's that the channels that are there are now conducting more calcium. And why that is still being investigated. So perhaps that same mechanism is occurring here, and it's sort of advancing whatever aging mechanism that is driving this signaling cascade. Next, obviously, we looked at ER calcium signals and even resting calcium levels. So this is work John McDade, postdoc in the lab. We always look at resting calcium levels. We're doing 2-photon calcium imaging, so we're not getting a proper ratiometric output. But basal resting fluorescence levels are always pretty much within the same range. Well, clearly here there's an increase in the resting basal calcium levels after these TBI neurons, in the neurons subjected to either a single or a repeat TBI. But interestingly, and again, a point of divergence from the AD models is we're seeing a reduction in the ryanodine receptor evoked calcium release only in the repeat TBIs. And we're thinking perhaps this is reflecting some sort of sustained calcium leak from the ER, which is contributing to this increased resting levels, but relative reduction in ryanodine receptor evoked calcium release. And again, so these are all still mechanisms we're identifying and trying to piece together what would be the underlying mechanism, but the phenotype seems has been very clear. And because we know that calcium dyshomeostasis is tightly linked to the amyloid and the tau pathology in AD, and in TBI and the chronic traumatic encephalopathy, it's the tau pathology that's really a signature mark for brain injury, particularly sustained affective brain injuries. And here we're seeing, in green here is the immunolabeled phospho-tau species. And certainly in the repeat TBI case, you see this so super linear increase in the hyperphosphorylated tau, even relative to the single or the sham TBI. So this, I think, we're looking to what's the source for driving this. And these are previous studies we've also done. This is in a mouse model. So again, we see very close internal consistency between the mouse and the rats in these models, where is in the TBI, the TBI mice, we're seeing this rapid increase in the hyperphosphorylated tau. But if we treat these mice with dantrolene, that Ryanodex, the soluble version of dantrolene, we see a complete normalization, back to baseline levels of hyperphosphorylated tau. So again, strongly implicating that calcium release through the ryanodine channel as driving the phosphorylation cascade in these animals. And so we've done, additional studies looking at 30-day treatment or even 2 or 3 day treatment after the initial TBI, and we still see that same just complete amelioration of hyperphosphorylated tau, even on much shorter timescales or shorter treatment regimens. So that seems to be very, very tightly coupled between the two. So I was afraid I was going to go over time. So I was just going to end here, but sort of summarize the sort of similarities and differences that we're seeing between the Alzheimer's and the TBI models, both to try and build a mechanism and perhaps see what would be good or useful therapeutic strategies, since TBI often or has a higher incidence of leading to Alzheimer's disease. Can we target these same pathways? And certainly a lot of the histopathology and the synaptic pathology is similar. Where there seems to be these interesting divergences is in the calcium handling, but the net, I guess, summation is, pretty fundamental deficits in both resting calcium levels and the TBI, or these profound increases in ER calcium release in the AD animals, seems to be driving a lot of the tau pathology and histopathology in both of these models that we know is central to the disease. So with that, I wanted to thank very much the people that have really been on the ground doing all the intensive labor with this. Certainly John McDade and Clark Briggs were very involved in the patch clamp and the calcium signaling in these TBI models. Nikki Barrington, also, and Alyssa Littlefield with a lot of the histopathology involved. And then Sarah Mustale, who's been central in the autophagy and lysosome work as well. So, and then colleagues, so the Dorothy Kozlowski has been the leader in this field. So without her collaboration, this project really wouldn't have come to fruition. And so we recently, not too long ago, got a Department of Defense grant to support this work, as well as like Bob Marr with the human-induced neurons, and Dan Peterson, my other collaborator on this TBI project, who's the head of our Stem Cell and Regenerative Medicine Institute. So with that, I will be glad to take any questions and delve deeper into any particular facet that you would like. And I thank you for your attention.
Thanks, Grace, for a really nice and interesting talk. And there are plenty of questions in the chat box, which I'm going to relate to you now.
Okay.
Maybe with the one of the most fundamental ones from Lucia Nunez. Is the increased calcium a cause or a consequence of Alzheimer's disease? What do you think?
Okay, so very good question. If you're going to put it into one box or the other, I would put it as an upstream driver, so it would be more in the cause box. I don't think it's a singular point cause of the disease by any stretch, but it is upstream and it appears before all the other major features. So it's observable prior to the amyloid and the tau pathology, before the cognitive deficits kick in. And by normalizing the calcium, you can prevent this wide array of seemingly disparate phenotypes. Whereas you can clear tau, but that doesn't necessarily fix the synaptic pathology. Whereas if you normalize calcium, you fix or reduce the amyloid, the tau, the synaptic pathology, the oxidative stress. So I'm pointing to the calcium signaling deficits as an early upstream driver.
Okay, thanks. Antonio Villalobo asked, is there any evidence about the involvement of naturally occurring calmodulin mutants deregulating ion channels in neurodegenerative diseases?
Yeah, so I don't have, put this way, because of calmodulin's role in regulated ryanodine receptors, I would anticipate it being a signaling component, but I'm not aware of any direct calmodulin-based mechanisms in the disease.
Dem liegt's fern von den anderen.
Is that? I don't know if that was a sufficient answer, but I don't know if there's, like, if people are pointing to mutations or alterations in calmodulin structure or function.
Then Martin had a question and was asking whether rectifying calcium signaling in the human neurons completely normalizes the autophagy, or
Yes.
something else happening to autophagy in addition to the altered calcium signals.
Oh, I'm sorry. So it was the question if normalizing the calcium
The first
restores?
question is, yeah, does the rectifying the calcium signaling completely normalize autophagy?
Yes.
And then there was a following, oh yeah. So you don't think anything else is needed? Only the altered calcium signals are causing the autophagy defects.
It seems a little too pretty and elegant, right? Cells are never that clean. So are there, no doubt, right? Because many elements are regulating and controlling lysosome and autophagosome function. So I don't think it's singularly calcium, but you can normalize it by normalizing calcium. So it's at least sufficient to fix the global problem. And we've just looked at, the only element we've looked at is that autophagic flux, right? So there might be other aspects of autophagy, structure, function, maintenance that we haven't looked at that aren't being corrected. But at least in terms of that autophagic flux, that seems to be completely rectifying the turnover.
Yeah. Then Thomas Simon asked how the ryanodine receptor oxidation is affected when you do multiple traumatic brain injuries.
So that's something we definitely want to go into in a lot more detail. So earlier, 2 years ago or so, as part of a study with Andy Marks's group looking at the phosphorylation state of the ryanodine receptor in AD. And so there's clearly certain particular like PKA-dependent phosphorylation sites that were hyperphosphorylated in the AD brain. Certainly oxidation, phosphorylation, nitrosylation of the ryanodine receptor is driving a lot of its open probability and sensitivity. So that is something we want to look at as what's driving the ryanodine receptor, perhaps leak in the TBI. We need to recruit some more sophisticated protein biochemists for that. But we think that's largely what's initiating is the oxidative stress is more than sufficient to do that. That's been demonstrated very clearly that oxidative stress goes through the roof initially, but perhaps what's sustaining it is those modifications to the ryanodine receptor.
Okay, there's another question from Martin around the TBI model. So presumably in the TBI model, the neurons are dealing with increased calcium levels and excitotoxicity. So could it be that the effect of dantrolene might not indicate that ryanodine receptors are causative, but it just helps the neurons to cope with a little less calcium?
Sure, absolutely. And so with the TBI model, we're now invoking that voltage-gated calcium influx that we haven't seen before. So yes, perhaps it's just helping to maintain overall calcium homeostasis, perhaps reducing that sustained elevated level of cytosolic calcium. We're not seeing perhaps the increase in expression that we see with AD. So I don't think it's a one-to-one, but I think because ryanodine, normalizing ryanodine function clearly is having broader, profound effects on the cell, but I think it's perhaps a different general mechanism than in AD.
Then we've got a question regarding your human neurons. Do you have any big differences between neuronal firing in age-matched iPSC-derived neurons and isogenic iPSC-derived neurons?
So we haven't done the isogenic controls, but we have not seen major differences in like the basal firing properties and action potential properties. I think the only thing we were noticing was a slightly blunted sodium channel currents. But again, we need to look at that in more detail. But firing properties look the same: thresholds, action potential, kinetics. Those did not seem to be different.
Then Gert asks, how does increased ryanodine receptor-mediated calcium signaling result in increased A-beta 42 levels?
So good question. So that, and actually another group study, Ules et al., 2012, did more of the biochemistry around this. But they showed that particularly from ryanodine-mediated calcium release is upregulating BASE, the beta secretase. Beta secretase is the key enzyme that cleaves, or one of two, that cleaves APP into A beta. So if you're getting more base activity, you're getting more A beta 40, A beta 42 cleavage. So it's largely through that mechanism.
Okay, Gert was having a follow-on question asking whether the G secretase activity is increased or whether there's less degradation. So that was his speculation about the increased A-beta levels, whether that is a mechanism.
Right. No, so Bob Marr, I don't think he's on the call. One of the areas that he also is very interested in is clearance of A-beta using and mediated through insulin-degrading enzymes, IDE. And so certainly he did more work in human tissue, but there's definitely evidence or suggestions that clearance mechanisms are also impaired. And again, through that IDE clearance mechanism. But that's, after it's already forming. So I think it's sort of a double hit, perhaps, in that we're getting increased, at least in the cases where there's, the ryanodine calcium signaling, increasing cleavage, and then reduced clearance.
Then Anthony Gaglioni wants to know, does dantrolene selectively block post-translationally modified winding receptor 2, but not normal type 2 winding?
Yes. Absolutely. So this is definitely more of a complicated story that we spent a fair amount of time looking into. And this is also with some of the more like protein modification work with Andy and then others. But normally the structure with RYR2, there is a dantrolene binding site. Usually it's in this sort of closed off loop or zipper. But when the RYR2 becomes oxidized or phosphorylated, particularly at those PKA sites, the structural change now exposes that dantrolene binding site. So we work more at the physiological level, and the supporting evidence for that is in our normal, whether it's the wild type neurons from humans or the age non-AD neurons or the wild type non-AD mouse models, dantrolene or Ryanodex rarely has much of an effect. So it's not just uniformly suppressing RYR calcium release. It's not doing much of anything in our control animals, but what it's doing is normalizing the response in our Alzheimer's animals. And so to me, that also speaks to, under normal conditions, that dantrolene binding site isn't either exposed or accessible under control conditions, but in these disease stress conditions, whether it's either oxidized, phosphorylated, now that dantrolene binding site is open, put in dantrolene or Ryanodex, now we're seeing our normalizing effect. And it rarely goes like below that, it just basically goes back down to normal function. So that's where the idea for our therapeutic approaches and sort of developing novel compounds around that, since it was working as the perfect negative allosteric modulator, right, which is what you want.
Then I've got one question from Oksana, whether there are any hints towards the involvement of store-operated calcium entry?
I'm sorry, I just didn't hear you.
Hints towards the involvement of store-operated calcium entry?
Yes. So this is Ilya Bezbazvani has been spending more time in that space. We've in our attempts to at least make sure we're not invoking other calcium signaling channels or to see what other channels are involved, have played around with blocking store-operated calcium entry and then seeing if that reverses or prevents the increased RYR calcium release. That's as far as we've gone down that path. We haven't seen much evidence for that, but Ilya is working more at STIM2 levels, showing changes in STIM2 expression and function in AD. So, within, who knows specifically, but there does seem to be some evidence of STIM or I changes, but I don't have a really elegant answer, but I defer you to Ilya's papers.
And then finally, I've got a question. You've shown in one experiment with the human neurons that baflomycin didn't have any effect. Have you looked at the effect of lysosomal calcium stores or acidic calcium stores with some other drugs, whether there's any involvement?
So this is the work of my graduate student, Sarah Mustale. I was going to go show you more of those studies, but again, I didn't want to go over. So she's got quite a few studies. I'm just pulling this one up here. We're looking at baflomycin treatment in the human neurons and then the accumulation of phospho-tau as a result. And she's got quite a few other sort of ongoing or preliminary experiments actually showing that baflomycin, by blocking those vacuolar ATPase proton pumps on the lysosome results in, and this is just overnight treatment, so pretty profound increase in, say, that pathogenic phospho-tau accumulation. And if you notice in this baflomycin, it's not the typical phospho-tau that you're seeing in the processes. They seem to be clumped into these little organelles. So whether these are actually, the phospho-tau that's now accumulating in the autophagosomes because they're not being digested or in some other endosome body. But that's, and then we can, in the pH, I don't have this slide, but if you release ER calcium, say through caffeine, the pH of the lysosome becomes more alkaline. right? So that's sort of a direct, and what we think might be happening is that, that calcium, that excess calcium that we see released in the AD models is interfering perhaps with the calcium extrusion from the lysosomes. It's inhibiting proton uptake perhaps, and then it's the lysosome pH is becoming more alkaline. We can prevent this either with baflomycin or with dantrolene. So that seems to be a nice little coupled system in there in terms of what's regulating lysosomal pH, and it seems to be interfering with the bacular ATPase itself. So I don't know if I kind of went off on one of my tangents because we were excited about findings. I answered your question correctly, but.
If you're up for a couple more questions, there were some new ones coming in. One was, is how are the AD-related mutations, for example, APP, PSN1, PSN2, relating to the autophagosome accumulation? Would also upper E4 be related to increased autophagic fluxes?
Good question. I don't know. I don't know, right? Because the APP mutations, there's APP, PS1, PS2. That's a good question. I don't know. But we'll add it to the list. Sarah?
Then Gert had one more question. What's your view on having more active ryanodyne receptors? Would it be sufficient to drive AD itself? CF, CPVT mutated ryanodyne receptors, or do you need additional factors?
So that's also one of my pet experiments that I want to do is overexpress RYR2 and see what AD or neurological phenotypes emerge. And we haven't done it yet, right? Because that'd be the perfect counterbalanced experiment. I would certainly anticipate more histopathology, the phospho-tau, the increased APP cleavage. I don't think you need the mutant RYR like that. Actually, there's no mutations associated with AD in the ryanodine receptors. It seems to be more the post-translational modifications making it leakier and increased expression, but not necessarily a mutation per se.
And I think let's have this one as a final question, which is going again in a slightly different tangent to the other calcium release channel on the ER. The IP3 receptors are also phosphorylated in a pKa-dependent manner. What is known about IP3 receptors in AD and TBI?
Oh, so I started with IP3 receptors, and they were the first ER calcium channel to be implicated in AD. And this goes back to, I guess, to the 80s. There's a group looking at fibroblasts from AD patients and putting in bradykinin to stimulate that GQ signaling pathway and seeing increased calcium released in the AD fibroblasts. And then subsequent work both by Mark Mattson and then Frank LaFurla and then Ian Parker. So this is where I got started with my postdoc, expressing, IP3 or mutant presenilin, which is going to cause familial Alzheimer's disease in oocytes, and showing that the IP3 evoked calcium signal is, greatly increased in the cells expressing mutant presenilin 1. And then I said, that's great, but let's look at this in neurons from mouse models of Alzheimer's disease. So certainly the IP3 contribution is significant. It's more in neurons localized to the soma and the proximal dendrites. So it's certainly driving probably a lot of the somatic deficits. However, one thing we also found was if you photo release caged IP3 but block the ryanodine receptor, the amplitude or the magnitude of that IP3 calcium signal goes down. So a lot of that IP3 triggered calcium release is also invoking a ryanodine response in the AD neurons. And we're not seeing, and its expression is consistent with this, much of an IP3 response out in the synapses, in the distal dendrites. And I'm interested also in the learning and memory component. So that part of the neurons is central to that. So it's certainly involved. presenilin, it was in one of the earlier slides, is also localized to the ER membrane, which is kind of odd because most of its substrates are plasma membrane. But the presenilin modulates the IP3 receptor and the ryanodine receptor. So certainly mutant presenilin is thought to be defective in that capacity.
Thanks a lot for that. There was one more question sneaking itself in whilst you were talking, and I promise that's the last one. Have you looked at brain inflammation in your models? Cells other than neurons, like glial cells, may have profound alterations too when calcium signaling is modulated. What are your thoughts on that?
Yes. So that has been up until recently, left out of the equation. Although clearly astrocytes and microglia are essential. So we are starting to look at that now in detail. So 2 of my students are specifically focused on those questions. So Sean Schrank and Nikki Barrington will be looking into that in more detail. There's other groups that have given this its proper respect years ago. Clearly, calcium signaling in microglia is affected. We know that, and astrocytic calcium signaling is also upregulated, and works by Olga Garashok have shown that in the AD brain, these astrocytes are just sort of randomly firing off across these networks in the in vivo studies that she's done. What that means isn't exactly clear, but you need those astrocytes to regulate sort of that trisynaptic circuit to take up a lot of excess neurotransmitter, calcium. It's critical in maintaining overall network homeostasis. And obviously neuroinflammation is one of the key features of Alzheimer's and neurodegenerative disease, certainly in brain injury. And those calcium signals we know are altered. I know less about the sources and the causes, but again, that's what we'll be looking at in the future. But no doubt we've been sort of ignoring 2/3 of the really important components in neurological disorders.
Thanks a lot for that. And I think I would suggest we finish the official talk now, or the official question session. Thanks to everybody for attending. I just want to point out, next seminar will be in 2 weeks on the 9th of July by Natalia Prevatskaya on oncochannelopathies, the calcium link to cancer. And right now, like we've done over the last weeks, we're going to turn the recording off. And if you actually want to turn your mic on, Grace has kindly agreed to stay on for a little bit. So if you want to ask her something directly, I know